Hello everyone. We'll be discussing some basic facts about tissue culture in our class today. We have divided the entire topic of our syllabus into four different parts. So these four parts will be dealt with in four different lectures so that each lecture gives you a crisp information about each of the topics. Now, plant tissue culture, as we all know, it's a method basically of culturing an entire plant from small cells or small tissues that are derived from the plant. Now, what these tissues are, where these tissues can be derived from, and what are the requisites with the help of which we can help this small tissue, a small group of cells, divide and multiply into a complete plant is what we are going to look at under plant tissue culture. Now, to begin with, the first thing that we are going to discuss in this lecture would be the basics of plant tissue culture, starting first with the theory of plant tissue culture, which begins with a small point of totipotency. Now, what is totipotency? Totipotency is basically the potential of a single cell to generate a total plant. So total plant generation, the potential to generate a total plant is totipotency of a single cell. And this term of totipotency was first, let us say, published by Heberland, who was studying the wound healing mechanism of plants with the help of certain loose tissues, which is called as callus. Now, in the due course of our study, we will be understanding that callus is a mass of undifferentiated cells. They are naturally formed by the plant during the process of wound healing. Now, for totipotency, for this process or for this concept of totipotency to become real in case of plants, there are two steps or two mechanisms that are critical. The two mechanisms that are critical in order for a plant to show totipotency is dedifferentiation and redifferentiation. Now, dedifferentiation and redifferentiation both take place for a cell that is already specialized. Now let us consider a cell which is present in the leaf. A leaf cell is specialized to perform the functions of photosynthesis. Say for example a root cell. A root cell, on the other hand, is specialized to perform the functions of root. For example, it will absorb minerals and water from the soil. A cell of the leaf, on the other hand, will be specialized to perform functions like photosynthesis. Now, both these cells are, even if they belong to the same plant, they are specialized in their respective functions. Now, let us take the example of a third cell, let us say an embryonic cell. An embryonic cell is able to perform all the basic functions, or let us say the household or the housekeeping mechanisms of the cell, but it is not specialized for any task. It is an unspecialized cell. So, this embryonic cell, which is unspecialized, it has the ability to generate an entire plant. Now, if I say that I can take any tissue from the plant, any cell from the plant, and I can make the entire plant from it, the first thing that I have to do is I have to take this, suppose I take cells from a leaf. I take this leaf cell, the first cell for this leaf cell will be to become an embryonic cell, to become an unspecialized cell. And then it can go on, it can go further and become other cells or other organs that are required to form an entire plant. So, the first step in a plant to become totipotent is becoming undifferentiated or becoming unspecialized from a specialized state. So, for totipotency, the first step is called as dedifferentiation. Dedifferentiation meaning a process where a specialized cell rewinds or backtracks from its specialization and again becomes unspecialized or again becomes embryonic in its basic routine. Now this unspecialized cell in the next step will redifferentiate. Redifferentiate meaning in this undifferentiated cell will now form the entire plant, meaning it will again form leaf cells, stem cells, root cells, etc. So it will again be specialized into different branches of the plant requirement. So it will again be differentiated, or it will be re-differentiated. So for a cell, for any cell of the plant to be totipotent, the first thing that it will need to do is de-differentiate from its original state of specification to an embryonic cell, and then again get differentiated or get re-differentiated to form the entire plant. Now there are two very important things that we need to remember at this stage. These two important things are, the first thing is that this process of totipotency is a very plant concept. Although we say that animal cells are also totipotent, but we must remember that animal cells do not have the potential to de-differentiate. De-differentiation is an extremely difficult and not a natural process in case of animal cells. It is a plant cell process. It's a process that comes very naturally and very typically to plant cells. The second point that I want you to remember is that why is this de-differentiation, or why is this process of totipotency such a natural mechanism in case of plants? Now imagine that a herbivore comes and eats up a few leaves of the plant. Now even if some parts of the plant are lost during herbivory, the same part of the plant can be regenerated. Now because the plant is a sedentary organism, it cannot move from its place, it has derived, or it has developed a tendency, or it has developed a nature to generate its organ or regenerate its organ very easily and very naturally. Because organ loss is also a very natural process for plants. On the contrary, if you look at an animal, suppose an animal is hunted by its predator. Suppose a tiger eats up a cow. Some parts of the cow, suppose two legs are eaten up. You cannot imagine the two legs of that cow to regenerate, because regeneration is not a mechanism that comes naturally to animals. Organ regeneration is something that is very inherent to plants, and because it is inherent to plants, we understand that totipotency also is inherent to plants, because totipotency naturally is a tendency of the plant to regenerate. Now, once we have understood this concept of totipotency, this is the basic idea behind the generation of an entire plant in vitro. In vitro meaning we use this concept of totipotency to generate total plants or to develop total plants in laboratory conditions. So this is the first part of our lecture. This is the first part of our topic, the topic 1. And with this we wrap up the first part of our lecture, which would deal with the plant tissue culture.